So we will move on to our next speaker. Hsiao-Lin Wang is an NIEHS postdoctoral fellow in the laboratory of Victor Corsis at Emory University in Atlanta. Welcome, Dr. Wang. She's also going to speak to sevoflurane in a mouse model.
Hi, thank you for Joe and the meeting organizer for giving me this opportunity to share my work. So I hope you can see my screen. Okay, so today I'll be talking about our work on how sevoflurane induces epigenetic alterations in the germ cell that result in autism-like behavior in our mouse model. So thank you for Dr. Totorovic and Martiniak for giving such a wonderful introductions and both clinical and mouse model studies. So I will be very brief on our introductions in terms of the previous evidence. But we first got interested, and we're interested in general how the neurodevelopmental diseases are inherited. Because a lot of the neurodevelopmental diseases, particularly autism, genetic cannot account for all the cases of autism. And based on the most recent report, genetic may only account for at most 30%, leading to a suggestions of what are other possible causes of such a high prevalence of autism, which is found in 1 in every 54 children based on one of the most recent CDC studies. So I highlighted the more high confidence genes identified by recent GWAS studies in the autistic patients. And as you can see that if this wheel highlights the entire chromosome or the DNA molecules in a human beings, almost every spot in the chromosome is covered with the autistic potential genes. But obviously, I don't think not every genes could cause autism. So, but many of these genes are implicated in synaptic functions, neurodevelopmental functions, and particularly chromatin remodeling pathways. So to answer some of the questions of how non-genetic effect or causes of autism, we use a mouse model, which we expose a mouse model with sevoflurane as both a presentation before addressed sevoflurane is one of the most commonly used volatile anesthetic agents, so that became the first choice of our exposure agent. So instead of exposing postnatally, we chose to expose prenatally both for 7 days in our mouse model 1, or expose for 1 day at embryonic day 12. And the reason we chose this window of exposure is to target the germline reprogramming stage. So we expose the pregnant females, which in the same time exposing the embryo in utero, as well as the germ cell that's undergoing reprogramming of the embryos. And it's important to note that in this time frame, that not only the germ cell is going through reprogramming, the cortex of the embryo that's in utero is also undergoing rapid neurodifferentiation, including neurogenesis and followed by gliogenesis. So I like to stress that I use the male germline transmission model. So I follow the male germline transmission by crossing the exposed embryo F1 with a control female, and then making a transgenerational mouse of F2 and the F3 intergenerational mouse. And then because we use a male germline transmission model, I follow mostly on the male animal. And then looking at the F2 intergenerational crosses and as well as the F3 transgenerational mouse, and look at their the sperm epigenome, the brain phenotype, and the behavior phenotypes. So first, We found that by exposing the animal to sevoflurane for 2 hours for just one day, the F1 embryo, which was in utero at the time, have a 40% of socially abnormal behaviors. So we use a three-chamber sociability test, which by testing how the mouse interacting with either an object or another mouse. So by looking at the F2, which are progenies of the germline that were exposed in utero, and we found 30% of the male animals are socially abnormal. And so we're interested in how this autistic phenotype could be transmitted transgenerationally, meaning that the animal that has never been exposed, nor the germline that give rise to them has never been exposed to the sevoflurane. And we found that out of the animal that we tested, it's around 40% of the male animals are autistic-like. So I think this percentages is not to a surprise because we would not assume that sevoflurane will cause 100% of autistic behavior in their progeny, both intergenerationally or transgenerationally. And then so we right now perform the three-chamber sociability test, but we're verifying the animal and the behavior using different autistic animal behavior tests, including the reciprocal social interactions or the repetitive behavior tests or the ultrasonic vocalization test, because one of the most key, strongest autistic patients are nonverbal. So question is then, if we show that our animals that are autistic, and then how are these alterations transmitted? One of our working model is that since we expose our animal during the primordial germ cell reprogramming window specifically, we think that in the inductions of the sevoflurane, when the germ cell is reprogramming, the chromatin is bookmarked by transcription factor, which is bound by transcription factor. So in the reestablishment of the sex-specific germ cells, these bookmarked regions cannot be remethylated. Hence, this is how the information is passed to the next generation through the germ cells. In order to measure the differences in transcription factor binding, we use the assay of transpose accessible chromatin using sequencing the ATAC-seq. So briefly is that we use an enzyme called the TM5, which will cut around the occupied regions. By having this enzymatic reaction, we can capture pieces of the chromatin or the DNA that is either covered or not covered by a protein or nucleosome. So after whole genomes, after sequencing of these samples, we can profile the regions that are either covered by transcription factors or the mononucleosome regions or regions that are covered by multiple nucleosomes. So to examine the epigenome and how the accessibility is affected in the sperm of the F1 mouse that lead to the F2 autistic animal, we take the mature sperm and then perform a ATAC-seq, which look at the differential accessibility and predict what transcription factor is found in the sperm that lead to autistic animals. And we found that there's roughly 1,500 of differential accessible sites, either gained or lost, and many of them are found at key autistic genes. And many of these sites are located in the intronic regions and intergenic regions, which are typically the regulatory regions of coding genes, and bound by various transcription factor and specific transcription factor. One of them is the CTCF, which is a zinc finger proteins that are master weaver of the genome in controlling 3D genome organizations. And we found, interestingly, that many of these regulatory regions that are bound or change their accessibility of how they are bound by transcription factor are actually localized in genes annotated for mental disorder for the gained accessible site, meaning for those sites that you have the increase of transcription factor binding, or in the lost accessible sites, meaning the regions that typically were bound by transcription factor, but in the presence or induction by sevoflurane in the sperm is no longer bound as much by transcription factor. And those are located near mental disorders, genes, or nervous system genes. We also look at the F2s of the sperm of the F2 animals, which will be the sperm give rise to the first transgenerational batch of animals. And many of these differential sites that we think are bookmarking for the epigenetic alterations induced by sevoflurane are found overlap between the F1 sperm and F2 sperm. And I look at the sites that are found in the sperm of F1 and as well as the autistic sperm of F2 animals. And many of these sites are located, and here are the example of the sites that are persistent between F1 and F2 generation of the animal in their sperm. They're located at autistic genes and neurodevelopmental genes, including the gene encoded for the calcium voltage gated channel, subunit alpha 1E, or the a very high prevalent autistic gene, the ARD1B, which is a Swiss-Smith chromatin remodeling complexes, or another gene, MTA1, which is part of the nucleosome remodeling complexes. So again, this is looking at the sperm of either the autistic animals or the sperm derived from the sevoflurane exposed germ cells. So, which they could be a potential bookmarking in how the sevoflurane induced epigenetic alterations are inherited or passed on to the progenies. So, but then how about, so what are the sort of the neurological phenotypes of these animals besides the epigenome of the sperm and the behavior phenotype of the sperm? So in order to address that, I looked at the F2 autistic animal. And again, F2 is the intergenerational generation in our animal model. And I look at the sensory area of the cortical plate in the adult animal brain and stained for excitatory projection neurons. And one of them is I use the FOXP2, which typically stains for the layer, like in the control cases here, typically stains for the layer 5 neurons. And then it is not present in the upper layer of the neurons. But to our surprise that, and it's quite exciting that the FOXP2 neuron not only stains in where it's supposed to be at the layer 6, but it also largely present at the upper layers. And again, I'm looking at FOXP2 is known to co-stain with the glutamine receptors, so is typically classified as excitatory cortical neuron instead of the GABAergic inhibitory cortical neurons. And FOXP2 is a transcription factor. So then that prompted us to think, okay, is FOXP2 one of these bookmarking? Is it possible that FOXP2's binding to the chromatin or its related pathway is how the sevoflurane-induced phenotype being transmitted in the germ cell and into the progenies of the animals? So I looked at the RNA-seq data in different stages of germ cell development, including from the primordial germ cell, which is when the embryos, the germ cell of the embryo is being exposed to, and then as well as the spermatogonian stem cells, as well as the mature sperm. And FOXP2 using clustering, FOXP2 is only found expressed in the primordial germ cell stage, but not in other stages of germ cells. FOXP2 bind to this T-reached motif that's actually bound by different types of FOXP or FOX proteins. So it's possible that the original information is retained by FOXP2 binding to a subset of an genome, and then by having different FOX protein expressed in different stages of the germ cells, the information is maintained by a sort of a group of an army of the FOXP protein to maintain this memory. So in addition to looking at the sort of the cortical lamination, which is how the projection neurons are located in the adult cortex, one of the human clinical phenotype is that the autistic children are either having a larger brain mostly, or sometimes have a smaller brain. So then looking at the F3, which is the transgenerational animals, and we found that first of all, the F3 animal compared to the control in red is weight less, and the weight of the brain itself is actually less. And here's a picture of the control and the F3 autistic brain. And as you can see, we tried to measure that the size of the brain is significantly reduced. So then I went down and measured different sort of parameters of the cortex and found that one of the lobes, especially the left lobes, are significantly smaller compared to control. But when we measure animals from the same lineage that's older, the adult animal of 2 months old, that the brain size no longer differ. And then the animal does not weigh differently from a control as well. And this is not necessary this match with what is observed in the autistic clinical patients, that many times after the patients are progressed into young adult, the differences in brain size no longer exist in autistic patients. So I hope I'm not going too fast because I know we're a little bit over time. So here I would like to just repeat in my conclusion is that we found about 30 to 40% of the F2, the intergenerational, or the F3 transgenerational animals, which are not exposed to sevoflurane directly, exhibit autistic-like behaviors. And there are many differential accessible sites found in the sperm of the animals. And then some of these sites are located near, in, and or around the regulatory regions of autistic genes. And maybe this autistic phenotype exhibited in the animal might due to the cortical neural migration defect in the adult cortex, preceded by the alterations in neocortical sites. And this is the acknowledgement. So I'd like to thank Victor for always the valuable suggestions and advice, as well as Joe, who bring this very important problem to our attention, and especially to members of Force's lab, especially Samantha Forster, who single-handedly did all the behavior work, and other members of the lab and funding agencies. Thank you.
Thank you so much, Dr. Wang. So glad you're doing this work. We have some questions, and then of course I have another question too. Dr. Shioda asks, can you comment on the sperm ATAC-seq data as to whether the accessible chromatin regions have significant overlaps with previously reported regions enriched with residual histones, or they are within supposedly protamine protected areas?
So for most of the regions we're looking at actually are not protamine bound regions. So I'm not sure about the overlap with the residual histone or the residual like the H2AZ that remains, like the bull marking like data. So, but that would be a very interesting thing to look into for sure. Thank you.
From Jessica Preston, great presentation. Did you perform the sperm ATAC-seq on frozen or fresh sperm cells? If frozen, how are the cells preserved?
Yeah, so we perform ATAC-seq on the fresh sperm using the stream-up protocol. So then we are retaining about 99% of pure sperms. And we also perform a tech-seq, although the tech-seq is not sort of as efficient in the frozen sperm. But instead of flash freeze, we use a cryopreservation. And we're happy to share the protocol. So please email me, and then I'm happy to share the protocol of how we perform a tech-seq in the sperm. So I don't want to bore everyone about the technical detail. I can go on with hours. So yeah, thank you.
All right, and Dr. Kors has actually responded to the first question. All ATAC-seq accessible regions corresponding to bound TFs are flanked by 2 well-positioned nucleosomes.
Yeah, thank you.
Dr. Mansi has a question. Great data, many thanks. I wonder if you could speculate on how you think changes in chromatin accessibility in sperm are maintained throughout development, multiple cycles of mitosis in the progeny to be present in their own sperm.
Yeah, I think that is a burning question that unfortunately we don't have an answer, especially around the model of sevoflurane exposed animal model. So we like to think that perhaps, or I like to speculate that perhaps by sort of having a collaborative network in different stages, because transcription factor normally does not constitutively express in different cell types throughout development. So through that network, and that's how the information is preserved, you know. And because at the end, we still found the same site at the sperm of the father of the animal, but also the sperm of that animal. So how it got there, that's a very interesting question. And our lab has a very nice report on how this information is retained using a bisphenol A exposed model that Victor talked about. And then in that, we have a specific example of how that effect is passed on. But I'm way too early to comment how sevoflurane is, you know, doing things that we're observing now. But thank you for the question.
Great. I have one quick question. So your model relied on a very specific timing for the exposure, right? And we've heard about other models where there was a neonatal exposure or even a later in life exposure to the sire. What particular effect do you think it has to expose during that very small in utero window? Of course, you can't speculate, but if you exposed later, would you see a different pattern?
Yeah, I think I definitely, that's a great question, first of all. I think exposing in different window will give us different pattern. But so we chose to expose this precise window just because we know germ cell is very vulnerable at that stage. So, but I think exposed later and based on data I have, not exposing to sevoflurane, that the effect may or may not sustain. And also exposing prenatally versus postnatally, it's a present a different sort of set of questions, you know. So I think exposing, and if I'm primarily talking about males, exposing males during sort of the maturations of the sperm, which then is the beginning of meiosis. I think that, and then I'm actually going to overlap with how much I found in what Dr. Maniak published, and then how that overlap, because he used a mature, sort of adult postnatal exposure, and that should partially answer some of the questions. Do they pose the same sort of threat in the germ cells versus, yeah. So okay. Thank you.
In post-perinatal brain, the NK